Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have James J. Phelan. He's a postdoctoral research fellow at Trinity College, Dublin. We're going to talk about, various topics: tuberculosis, infectious disease, and cellular metabolism. So, James, thanks for coming.
Thank you, Richard. Thanks for inviting me on.
Yeah. Well, tell me about your research. What are you working on?
Well, it's very, I suppose, expanse in terms of what I do. I suppose my expertise will be in cellular metabolism, really, and I would have started out doing and exploring cellular metabolism in, I suppose, the early part of my career in the context of cancer, specifically esophageal cancer. And while looking at esophageal cancer, one of the main risk factors for looking at esophageal cancer is a condition called Barrett's esophagus, which is effectively a distortion of the cells there, and this is the biggest risk factor for esophageal cancer. And I was studying different parameters, particularly cellular metabolism, and how cellular metabolism changes, and it makes individuals more susceptible to actually developing esophageal cancer.
You said the number one predictor or precursor for esophageal cancer is what? You gave some acronym.
Yeah, so it's called Barrett's esophagus, or intestinal metaplasia. So it's an inflammatory condition. So I suppose another way to put it is if you can imagine someone has a hot drink and you burn yourself, if you like, this could be potentially lead to, in the short term at least, to esophagitis. And over time, people might have issues with their stomach, and they might get reflux from their stomach. And these cells at the distal part or the bottom part of their esophagus gets chronically exposed to bile salts from the acidic stomach. And over time, these normal cells will differentiate and actually change into different cellular phenotypes. One of them being Barrett's esophagus. And then over time, this condition called Barrett's esophagus, if exposed to subsequent insults and stress, will actually turn into dysplasia, which is it's even closer to cancer again. And then again, over a longer period of time, again, this can actually transform into cancer. And sometimes it is a progression. It's a sequence of events from Barrett's to low grade, high grade, and then finally to adenocarcinoma or esophageal cancer. And I suppose what I was doing is I was characterizing cellular metabolism along this sequence. And I did this for about 4 years, and this resulted me into. I subsequently followed this up into infectious diseases. So I moved from a cancer field into an infectious disease field. But also at the same time, I also jumped into looking at immunology, and it was actually a contrasting role, because I suppose in the cancer field, as you know, various cancer patients are burdened with high inflammation, and the goal with therapeutics especially would be to reduce the inflammation associated with these diseases, and particularly with the likes of tuberculosis, which is one of my specialities. It's TB, particularly late stage TB. It's characterized by a huge inflammatory burden, similar like what's been reported with COVID-19 at the moment.
Well, so there's a lot to talk about. The 4 years you spent characterizing the metabolic changes. Can we talk about that first? What happened throughout all these stages of progression to esophageal cancer? What did you notice?
Well, I noticed what we did is, because about over a decade ago, not many people were actually studying what we term as the field calls it, cellular metabolism. And I suppose it's hard to actually pinpoint and define cellular metabolism. But the best way to put it is that, I suppose, if you can think of various vehicles, if you like, on the road, they all have all these different cars on the road. You might have a car, an ambulance, you might have a truck, a lorry, you'll have a digger. You have different vehicles that have different functions. And that's like an immune cell or a cell in the body anywhere. But the thing that actually connects all these, and what's the most common part of them, is the way they metabolize their glucose, i.e. their cellular metabolism. And I suppose if you like to make things a smaller, a little bit more complex, is that if you have particular cells that have two metabolic pathways, look at it as a hybrid car. And what I was doing was I was actually characterizing the different types of metabolism, or what and how much these cells use particular types of metabolism. So, for example, I found that as patients progress from Barrett's metaplasia, or this inflammatory esophageal condition, to cancer, they used this aggressive form of metabolism called glycolysis more. So they did. And glycolysis, this metabolic pathway called glycolysis, is actually associated with higher flux of cytokines and chemokines, and which is damaging to the tissue and obviously to the patient then. And that is our main finding.
The main way that cells process first is what oxidative phosphorylation.
Yes.
And glycolysis is usually a cancer-specific form of metabolism.
Exactly. Yeah. Absolutely. Got it spot on, Richard. Yeah.
But just quickly, what's the trade-offs of the two methods? Why do you think cells switch?
Well, the best thing about glycolysis is that the cancers need energy. They need energy to grow, to differentiate as fast as possible. And glycolysis, and switching to glycolysis, termed the Warburg effect, this fancy term used to actually define it, switching from oxidative phosphorylation to the other metabolic pathway, the glycolysis. It does this because glycolysis is a lot more, I suppose, efficient. And I wouldn't say efficient, it's much faster to produce energy, metabolic or cellular energy, than oxidative phosphorylation, even though oxidative phosphorylation is a lot more efficient at doing so. Glycolysis does it much, much faster. And I suppose you might have heard it in the news, your listeners might have heard it in the news about this so-called ketogenic diets, particularly for cancer patients, as a potential adjuvant therapy, not as a preventative treatment or even a cure, but just as a means to actually help the burden in certain patients. So what these ketogenic diets are based on are they bypass glycolysis. So they're given ketone bodies, and then these ketone bodies are they literally bypass glycolysis and instead go straight into the oxidative metabolism, and therefore the cancers can't actually use these ketone bodies. So you're effectively starving your tumor of these intermediates that are generally produced from glycolysis, effectively, and like even a decade ago, these ketogenic diets were proposed as a potential adjuvant therapy. And to this day, they still are.
Have they been found to be effective first before you describe them?
They are found effective, but they're never implemented. And they're really only going to be implemented, in my opinion, as an adjuvant therapy. You know, they do help, but they're not curative, if you know what I mean. They're not prophylactic. They don't prevent the disease. Yeah.
Are there people that faithfully do them, and how much of an effect do they get, or they only do them in combination with other activities?
It's generally in combination with other compounds, other chemotherapeutics, and that sort of thing. Yeah.
What's the observation? How helpful are they?
They help. They generally, and sadly, they're used in late stage as well for patients. Those patients who are very uncomfortable, they're used then as well. There's been reports that patients have found it to feel better with using this. But I suppose more significantly, this work that I was doing was, I would have finished that about going on 6 years ago now. And since then, I've been using that, and I've been applying what I've known with cellular metabolism in the context of infectious diseases. And in that model, and TB, I think you talked to one of my colleagues a few months ago and about TB and what our work kind of does. But what our our lab is, we looked at infecting macrophages, and we look at infecting macrophages. Macrophages run immune cells of the body, as you know, and they're the first line of defense during TB infection. So if an individual gets infected with TB, the macrophage tries to clear the infection. The macrophage immediately increases its glycolysis, or its rate of glycolysis, to help it get rid of infection. Because if we come along and we inhibit glycolysis in that macrophage, the TB overwhelms the macrophage. So it does. So what we've been doing in our lab is unique in a way, because various research institutes globally, what they do, and they've described it as late stage TB is characterized by this inflammatory burden. And you don't really want to increase glycolysis in these particular patients, because if you do, then you're going to get higher levels of inflammation, which is a no-no. But we hypothesize in our lab that if you can turn on glycolysis during the early stages of infection, you could potentially actually switch on glycolysis and therefore help eradicate the infection. And this is the kind of work that we do at the moment. So it's turning what I did during my PhD and in the cancer field on its head, really, where in the cancer, you don't want to switch on glycolysis, whereas during the early stages of TB, you kind of do. We actually found, and my research actually recently published this year, actually using what we did was we found that an iron binder, something called desferrioxamine, which is currently used in the clinic as an iron chelator, and it's actually currently in clinical trials for COVID-19 as well. But what we found is this compound can switch on glycolysis is in macrophages infected with TB. And can actually help clear the infection as well. But effectively, we're in the middle of actually looking to see if the effects and this iron binder can actually help in combination with current antibiotics that are used to treat TB as well. We showed that by simply using this compound, you can switch on glycolysis, you can up the levels of various different cytokines as well. So these cytokines, even though they might be pro-inflammatory and they might be harmful in late stage TB, we know that they're actually protective, and they actually help kill TB in those early stages of infection. So they are, which was surprising. Even before I started in the infectious disease field, I would never have thought that. So, and that's only 5-6 years ago now. So that's probably one of the kind of surprising elements to that my research in the early days, at least anyway.
Is there another way for respiration to happen besides oxphos and glycolysis, or is it only those two that's understood?
Yeah, good question. There are other metabolic pathways for sure. There are definitely other metabolic pathways, and I think even though people have been looking at metabolism for 10 odd years, we've only really scraped the surface of what they really do. I think most people at the moment are looking at oxidative phosphorylation, glycolysis. There are others. There's lipid metabolism. People are starting to look at lipid metabolism now. Other people are looking at other pathways that jut off glycolysis, such as the pentose phosphate pathway. And there are even certain amino acid pathways as well, tryptophan pathways, glutamine pathways, that people are starting to investigate. Because, for example, glutamine, or downstream from glutamine, you can get the production of intermediate called succinate. And one of our professors in our university is doing great work with succinate and itaconate, which they've shown and others have shown to be pro-inflammatory and anti-inflammatory respectively. So them alone. It's very surprising, because even 10 years ago, to think that a cellular intermediate such as itaconate or succinate could actually exhibit anti-inflammatory or pro-inflammatory effects respectively, it's awesome, because this could pave the way for potential, just firstly for understanding how the cell works, but also for therapies. For example, itaconate, which is shown to be anti-inflammatory, if you can put that on a macrophage or an immune cell, you could actually help it reduce its inflammation, again potentially in those patients who don't or to have unwanted levels or high levels of cytokines flux in that,
So it's only at certain points you'd want to intervene and lower the inflammation, right? Exactly. Or do you want to chronically lower it?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You do at a certain point. Yeah, I think that the key is a balance. I think the early stages you need a kind of a certain flavor of macrophage, if you like, that has a balance between promoting these pathways but also damping them down. I would argue myself, maybe from coming from a cancer background, you want to switch these off late stage. You want to get out your anti-inflammatories or your anti-metabolics even, and you want to switch off these pathways. But it's very difficult because cancers and cancer cells and normal cells, they both have oxidative phosphorylation and glycolytic metabolic pathways working concurrently. And it's very hard to target these specifically because various different compounds, they don't specifically bind to the ketone bodies. Now, for example, as we mentioned, that is actually good because normal cells don't really use ketone bodies. So because they have their metabolic machinery intact. So it's the cancer cells that have no choice but to they won't be able to
use glycolysis.
Cancer cells generally, use glycolysis,
But they have to. You said before that, do cancer cells use oxphos, or they only use glycolysis? Or is it not so clear cut? So I think most cells, if not all cells, will use both pathways, but glycolysis is commonly associated with cancer cells. There will be a baseline levels of oxidative phosphorylation, absolutely, yeah. So it sounds like as you progress towards cancer, at least esophageal, there's a recurring persistent inflammation.
Yeah.
So it's good to reduce it at that point. And then in late stage cancer, there's a reoccurrence of it now in a nasty form with cytokines, et cetera, that kills the person. So there you want to suppress it. So those are the 2 points that you want to intervene.
Absolutely, yeah. Exactly, yeah. And trying to determine that switch point, that that seesaw moment where it flips from one to the next, it's very difficult. It's very, very hard to determine that juncture, if you know what I mean. But you got to spot on exactly.
So what? All this has translated into infectious diseases. So let's talk about that. Like in the context of TB, what happens to someone? How does the disease progress? And then when does the inflammation component come in?
Well, it's mainly, you have early infection. So individuals who get infected can either clear it, they can either, they can kind of clear it and retain it and they become latent, the infected, or they can go on to progress to active TB disease. Latent infection can be associated with granulomas in the lungs, but also active TB disease. You'll have granulomas, you'll have cavities, you'll have an influx of immune cells that are spewing out cytokines and chemokines. It's thought that individuals who are immunocompromised, while the immune system is intact, it's keeping down and keeping TB under control. But individuals who are immunocompromised, they no longer can actually dampen down the TB onslaught, if you like. So it reacts, if you like. So, and that would be the case in individuals who have latent TB, for example. But the exact triggers, that's still undetermined.
Well, have you looked at other cancers, for instance, other infectious diseases? Like I would think inflammation is probably common to many, many diseases, many cancers as well. Absolutely,
yeah. So I suppose I've been, even during my research, I've been collaborating with various different PIs across the college here and abroad, and I've looked at, in terms of the cancers, we've looked at colorectal cancer. We've also looked at rheumatoid arthritis, which is an inflammatory disease of the knee joints. And it's very much cancer. Again, that inflammatory burden is all mediated by glycolysis. Again, you inhibit glycolysis, you don't have this cytokine influx. So, for example, one of the key cytokines across most cancer types and infectious diseases, particularly in a context of a macrophage model, would be a cytokine called IL-1 beta. So if you actually inhibit glycolysis during an infection or in a tissue, you'll get lower levels of IL-1 beta, telling you that metabolism is intrinsically linked to the production of these cytokines. And this seems to be the case from cell model to cell model in most cases across different disease entities.
So it is quite translatable across these models. And I think in some ways the cancer field has been ahead of the game with this. And in some ways, as a result, then now that I'm in infectious diseases, I'm learning a lot from other people and the great work that everyone is doing on this. So we're able to kind of ask different questions, build on their work and ask different questions. Because as I said, this area is only now being really understood. I mentioned succinate and itaconate and how they can affect pro-inflammatory and anti-inflammatory profiles. But there are hundreds and thousands of intermediates out there that people have to look at, again, in different contexts, in different infectious disease models as well. We even our lab in the next definitely our university and institute will be looking at COVID-19 in the next few months and years probably now. And these are certainly important questions to ask in the context of that too. And I've no doubt they will be asked. Not only by us though, but by various institutes globally,
Going back to cancer, why do the cells use like glycolysis versus oxphos? I thought it was partially because the mitochondria are damaged and the cell can only primarily do it that way. Or is it a preference instead, a choice?
Yeah, that's one of the reasons as well, because the mitochondria have an inefficient DNA repair capacity system. So if their mitochondrial DNA gets damaged, they can't repair it, and as a result, then they have to switch to glycolysis. But in my opinion, looking at this for so long, the main reason to switch is because they're just greedy. They want more energy, and glycolysis just will make them a lot more energy a lot faster, in my opinion. And you never know, particularly there's like certain research now is looking into the fact that, for example, TB can actually manipulate and damage the mitochondria in order to get cells to switch to glycolysis, particularly in the chronic stages of TB, because it can actually induce this higher glycolytic metabolism, and therefore it'll induce more damage, Well,
there's got to be trade-offs, otherwise cells would just do it all the time.
Oh yeah, for sure. Yeah.
So glycolysis enables production of energy faster, but there has to be a trade-off. There's something it doesn't do as well, I'm sure. Otherwise, it would be the preferred method of respiration.
Yes, that's an excellent question. The thing is, it doesn't really, you have a buildup of lactate. So I think you have huge waste. So you have the likes of, for example, the best way is when people exercise and you get a buildup of lactate. So there's huge waste products. And one of the things that we think that people are trying to do or hypothesize at the moment is that the byproducts, such as lactate, which is the end stage of glycolysis, lactate is being converted to certain pro-inflammatory markers, so intermediates that subsequently can actually promote further disease progression. So what's happening is, and in the context of TB, we think lactate is actually potentially feeding back on the system and actually helping to promote different other cellular processes such as autophagy, which is actually it effectively engulfs TB and kills it. But trade-offs are and I mentioned it was a good question because it's very, very, it's only starting to actually, people are only starting to ask that question. And one of the kind of main things that our lab is looking at is lactate, and particularly its role. It's not very known at the moment, but I think it's mainly down to their just need for getting sugars and getting more energy at the moment anyway.
Okay. So what's the next step? What's you're looking forward to now in the research? What are you trying to figure out? Or are you switching to COVID or, it sounds like there's a ton left to figure out. So what's next?
I suppose I won't be switching to COVID, but I know that the kind of next part of the research is, because a lot has been learned in that, and we found out a lot in the last 10 years, I suppose. And we've only really scratched the surface. And I think one of the big things, I suppose, at the moment is this topic as well. A very talented colleague of mine is looking at innate training and the role of metabolism in potentially helping innate cells, such as a macrophage, to actually expose them to an insult or a pathogen. So when they get obviously infected, if they get infected next time, that they're better capable of actually responding to that infection the following time, which could have potentially huge benefits down the line for the likes of host-directed therapies and vaccination strategies as well. Because we mentioned metabolism, but metabolism, various intermediates, such as I said, I mentioned succinate already and citrate or itaconate, but they're all alter host epigenetics, which is massive in terms of vaccination strategies and also in terms of host-directed therapies. And it's known that these can actually alter other transcription factors, such as HIF1 alpha, which is the main metabolic switch for switching on glycolysis. So that in itself would have a huge ramifications, especially for innate training, for example,
Okay, well, very good. What's the best way for people to find out more about your research and keep tabs?
If you go to our website, tcd.ie, and if you can type in my name, James Phelan, with a PH, and the profile is there, and all our lab is there as well. We've quite a big lab at the moment, past and present labs. My past lab was the Department of Surgery, so you can look up the Department of Surgery for my past research and the lab's past research, and then my current lab is the TB Immunology group in Trinity College as well.
Okay.
It's a very novel area at the moment, and a lot of people don't understand what's going on, but it's getting more exciting day by day.
Well, thank you for coming. I wanted to put this one or two questions back in. I forgot to ask you. During a cell's lifetime, in our body, how many mitochondria are in a cell? Just, I know, ballpark, it depends on the cell. And how often are they dividing and forming new mitochondria? And does that happen only when the cell, divides itself, or do they divide independently and, change numbers?
How many? So I suppose your first question was how many mitochondria in the cell. I suppose that literally can change from cell to cell in the same person. Really, that can literally change from cell to cell. To be honest,
ballpark.
I couldn't actually. I'm going to say 20. It could go up to 100. Depending on the metabolic needs of the cell, for example. So if a cell is infected, it's going to need a lot more energy. For example, if it was infected with a pathogen, it would need a lot more reactive oxygen species to potentially kill that pathogen. And reactive oxygen species is produced from the mitochondria, so it's going to lead to a lot more mitochondria. It depends on the stress of the cell and the microenvironment of the cell, really, for how many.
But are the mitochondria dividing independently of the cell itself, or they only divide when the cell divides?
No, the mitochondria actually divide, through fission and fusion events. So, for example, if a mitochondria gets damaged, what can happen is that the mitochondria can undergo fission, and they can actually divide, and then that gets rid of a defective. One of those mitochondria are defective because they're after getting rid of the defective part of the mitochondria, if you like. And then that undergoes a process of mitophagy to break down that defective mitochondria. And then that mitochondria that was remaining can actually get bigger then. So it can. But yeah, they can. So
they divide independently of the cell dividing. So the cell doesn't have to divide for the mitochondria to keep proliferating, right?
No, exactly. Yeah, exactly. And if there's a lot of mitochondria, they can actually fuse back again as well. Yeah.
Oh, really? They can fuse back into larger ones?
Yeah, they can fuse back into larger ones. Yeah, exactly. Yeah.
Okay, so when they are damaged and they go to a glycolysis preferred path, do they stop dividing? Do they divide? If they divide, do they create more damaged ones? Like, how does that change the dynamics?
I would hypothesize that they divide more, if anything, because if they are damaged, they're going to need to get rid of the components within their DNA that are actually damaged. So they're going to undergo a lot more fission in order to do that, to get rid of those defective mitochondria.
But wouldn't this predispose the cell to apoptosis? Because I would think the cell would have a monitoring system that says, we're getting a proliferation of mitochondria here, and we're getting all these molecules off of them that appear to show that they're damaged. It may be time to kill myself here, or they have apoptosis, or
yeah, absolutely, super questions. Absolutely, because I think we talked about fine balance. So, and this is the mitochondrial's fine balance. So if there's too much going on and the cell is burdened by too much stress and too much defective mitochondria, it does tip into the side of apoptosis. Absolutely. So this is so these processes of fission and fusion, these are happening prior to apoptosis. So if a cell does get burdened, it triggers apoptosis. Absolutely. This is mitochondrial mediated as well.
So is anyone studying the dynamics of the mitochondria in normal versus infected cells?
Our lab have actually started looking at, absolutely, yeah. I did mitochondrial work myself in the context of cancer, and we do see changes even from that initial Barrett's esophagus sequence to esophageal cancer. We published papers in the past looking at the different expression of different proteins, particularly DNA, and we looked at mitochondrial DNA, and we found that there were changes in mitochondrial DNA happening across this as they progressed to cancer. And we found that these DNA or these alterations in DNA were actually associated with alterations in mitochondrial membrane potential in the mitochondria, which are actually linked to the production of ROS and through different pro-inflammatory markers as well. So they are. So very much so. They do have profound effects. Mitochondrial alterations in mitochondrial function have profound effects downstream, certainly down as far as affecting the cell from a kind of an inflammatory flux point of view, if you know what I mean, like in terms of how it produces cytokines, and these cytokines more than likely also affect bystander cells as well. You know?
Okay, well, excellent, well, very good. Well, James, thank you for coming on the podcast. I appreciate it.
And thank you so much, Richard. Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.